everyone. Welcome to our MPRINT webinar series. I'm pleased to be back today with three of our support pool awardees from Vanderbilt University Medical Center. Dr. Anna Patrick, who will be discussing deciphering the genetic contributions to gene expression in juvenile idiopathic arthritis. Dr. James Antoon, who will be talking about his work with Oseltamivir and prevalence of concurrent medications with drug-drug interactions. And Ashley Leech, who will be speaking about late maternal death and overdose among pregnant individuals vulnerable to opioid use disorder and with opioid use disorder during pregnancy. So without further ado, I will welcome them to begin. And I don't know who's starting, so whoever would like to begin, go ahead and share your screen. Thanks. Anna, were you going to start today?
Yes, I am working on the sharing the screen part. I'm trying. Bear with me for 1 second. Okay, is what you're seeing correct?
No, it's the slide share or the speaker view now.
Okay, let me try one more time. Is what you were seeing correct?
That is correct. Great.
All right.
Thanks.
Thank you so much. All right, so I'm Anna Patrick. I'm a physician scientist in the Division of Pediatric Rheumatology at Vanderbilt. I'm going to talk about my support pool project, which was just kindly introduced. So first, juvenile idiopathic arthritis, or JI, is the most common autoimmune arthritis in children. It is diagnosed clinically by 3 criteria. Symptoms begin before the age of 16. The arthritis must be in 1 or more joints for at least 6 weeks. And this is a diagnosis of exclusion. So the arthritis cannot be due to malignancy, infection, or another source. So here I'm showing 2 pictures of children with JI, which highlights the wide range of disease severity. Shown on the right is a graph with patient number on the y-axis and age of diagnoses on the x-axis. Girls are the purple bars and boys are the blue bars. You can see that there is a peak age of onset before 5 years old. In diseases with an early age of diagnoses, like JI, it is suggested that there is a higher genetic contribution to disease. So JI is a lot more complicated than the prior slide, however. On the left, I'm showing the clinically defined subtypes of JI. Patients have a range in disease severity from mild to severe, varying chronicity and varying responses to therapeutics. On the right, I'm showing that we have targeted, powerful, numerous, and expensive therapeutics to treat JI. And some of these are very targeted, some of them are less specific. We have clinically defined subtypes and we have clinically defined therapeutics, but with our current knowledge, we do not know the optimal therapeutic for an individual patient. There's really a critical need for precision therapeutics in JI to fill this knowledge gap. One of the ways we have attempted to understand JI subtypes is through GWAS studies or genome-wide association studies. Thus far, the data has not provided the answer for how to prescribe therapeutics. Another approach is to use gene expression to understand the link between subtypes and therapeutics. So the focus of my project is understanding the genetic contribution to gene expression in JI. To do this, we will use a technique called PrediXcan, which allows us to use information from single nucleotide polymorphisms, or SNPs, to predict gene expression in different tissue types. Our hypothesis is that the genetic contribution to gene expression in early diagnoses JI is going to predispose to increased immune activation and exhibition of inflammatory cytokines. So we have 2 aims, and this is just our project experimental design. We will use JI patient and healthy control comparisons. In aim 1, we study the existent JI GWAS summary statistics with S-PrediXcan. In aim 2, we will study individual level JI genetic predictions from de-identified electronic health records, or EHRs, linked to our institution's DNA biorepository called BioView. So the goal of these two aims is to identify a genetically predicted JI pro-inflammatory phenotype. A key part of this project is the strong collaboration formed with colleagues in MPRINT and precision medicine. Elizabeth Jasper at VUMC is performing the PrediXcan and GWAS studies, and the VUMC phenotyping core is helping identify patients in the EHR. I'm only going to use one slide to describe the methods for PrediXcan and S-PrediXcan. The take-home message for this slide is that genotypes have been used to develop models to predict or to impute gene expression in a variety of tissues. PrediXcan then takes these imputed gene expression levels and conducts a test of association between the expression levels and your phenotype of interest. S-PrediXcan is a meta-analysis approach that conducts the PrediXcan test using genotype association summary statistics, rather than performing the tests on individual level data. In our study, we used summary statistics from GWAS studies. We also utilized existing models for 48 relevant tissues from the Genotype Tissue Expression Project and a unique placenta model that was developed by the Velez Edwards lab at VUMC. We tested the association between gene expression levels and JI in the 48 tissues and the placenta. So here I'm focusing on a GWAS study that used a high quality JI cohort, which came from the United Kingdom. So in this study, there were around 3,300 JI cases. These are a combination of all the JI subtypes, and then around 9,100 control cases, all that came from the UK population. And then the data type are the platforms they used to identify the SNPs. So we then performed S-PrediXcan on the summary statistics from this study. The S-PrediXcan model has many potential input SNPs. The first row shows the number of possible PrediXcan model SNPs that were included in our GWAS data set, which was excellent. It was 96%. Then we determined the Bonferroni corrected p-value for significance. And then we identified the significant genes for both total and unique genes. Total genes includes genes that are repeated as significant across multiple tissues. Unique genes removes any repeats. For example, if HLA-DRB6 is significant across 35 different tissues, then it would be counted 35 times in the total, but it would only be counted once in the unique genes. This is a Miami plot depicting the S-PrediXcan results. The study SNPs are below and the predicted gene expression is on the top with significance on the y-axis. The predicted gene expression is shown for specific tissue types, which are represented by the colors on the right. It's okay that you can't read the colors. It's okay. So we have a very dense set of enriched significance in chromosome 6, which is a known region important for immune genes, and it's highly associated with autoimmune conditions and with JI. In this slide, we've zoomed in on chromosome 6. You can see there are many significant SNPs below and significantly differentially expressed genes above. The multiple colors in the peaks highlight that many genes are significantly different in multiple different tissues. I want to highlight the top 6 significant unique genes along with the tissue type with the lowest p-value. It is important to note that HLA gene expression differences are identified, which would be predicted. We also have additional genes with roles in the immune system and others with less well-defined functions. So this is a really rich data set for further analyses. Finally, we performed pathway analysis on the significant genes and found enrichment of genes in immune system processes, including interferon gamma signaling and others. This result shows that the identified genetic contribution to gene expression in JI predicts differences in processes known to be important in the immune system. So now we're switching over just a little bit. So BioView is, and this is really to understand how we can obtain individual level JI data. So BioView is Vanderbilt's biobank of DNA extracted from discarded clinical blood specimens. BioView subjects are linked to a de-identified electronic health record. This enables researchers to access genetic data and DNA that's linked to a dense longitudinal EHR. You can see on the right-hand side of the screen, there are a large number of records in BioView, and there are a large number of samples that have been obtained. And additionally, at the bottom, over 100,000 samples already have genotyping or sequencing data. So this BioView resource enables exploration of a variety of research questions, including investigating the relationship among genetic among genetic variations, disease susceptibility, and variable drug responses in our JI population. So we have been building highly sensitive algorithms to identify JIA patients in the EHR that links to BioView. On the left depicts how our algorithm was developed. On the right, I'm just highlighting that multiple ICD codes are associated with JIA, and accurate identification of subjects is complicated. So with the help of the VUMC phenotyping core, we have been making these algorithms more portable. On the left, we used an algorithm requiring increasing counts of JI-associated ICD codes. We found that increasing the number of JI ICD codes required increased the positive predictive value, which is red, while decreasing the total number of JI cases identified, which is black. We then decided that we would include a requirement for 4 ICD code counts to be present to count a case as JI, and we did this for our next step. We added an age constraint for the first appearance of a JI-associated ICD code. And we did this because since this is a pediatric diagnosis disease, the age at which ICD codes begin to appear can really help you understand whether a patient truly has JI or not. So we found that addition of an age constraint significantly improves the positive predictive value, again, also in red. We found that as the age constraint was at a younger age, the total number of JI cases identified decreased. So we selected a JI algorithm requiring greater than or equal to 4 ICD code occurrences on separate days and the first appearance of an ICD code documented at or before 20 years. This algorithm identified 1,583 JI cases with a positive predictive value of 0.95. One of our goals was to leverage these algorithms to identify JI cases with genetic data available that links to the longitudinal patient records. So here we are taking the algorithm that we use to identify JI cases from the EHR, and on the left top is a very broad definition of JI, where you look for any chart that had any JI ICD code that appeared at least one, and we found how many different of these cases had BioView DNA available and how many of them already had MegaX array available. We then, if you look down below that on the left, we used the highly sensitive JI algorithm, where we had a more tightly and well-defined JI cohort, and then identified BioView DNA available and found that we had 242 cases that already had MegaX array data. And we need a comparison group. And so for our pediatric controls, we took cases from the EHR that contained at least one healthy well child check ICD code and found those cases that also had MegaX array available. And though I'm not discussing it here, we have requested and are in process of obtaining additional MegaX arrays from the highly sensitive JI algorithm BioView DNA that has not yet been studied. So to study the genetically predicted gene expression in our highly accurate BioView JI cohort, we first performed a GWAS study and applied the results in the PrediXcan pipeline. Dr. Jasper performed filtering and quality control of the MegaX array data. The final cohort characteristics are shown here. She then completed a genome-wide association study and PrediXcan of this data. So this GWAS was adjusted for 10 principal components, and we ended up finding 466 SNPs that are significant at a genome-wide level. 349 or 75% of these were on chromosome 6. Many of them are very close, associated with the same gene, or are in linkage disequilibrium, which is something we would have expected. Some are SNPs or genes that have already been associated with JI. And specifically, we have 2 SNPs already that were found in prior GWAS studies. So this is a really rich data set. When we look at S-PrediXcan from this study, we found 4 genes with differential expression in JI significant by Bonferroni corrected p-value. If you loosen your p-value constraint, of course, you begin finding additional genes. So this is a really nice study where we have extensive longitudinal data associated with these cases that we can now perform additional studies on. So in conclusion, PrediXcan identifies differences in the genetically predicted gene expression in JIA. We have identified an EHR-based JIA cohort appropriate for genetic studies aligned with longitudinal clinical data. And our future directions are much larger than our conclusions were. We want to validate the portable JIA algorithm across institutions, and we're planning to do this in the eMERGE network. We have additional mega X-arrays from the BioView JIA cases that we are obtaining. We are continuing our analysis of the GWAS and PrediXcan results, and we have a very rich data set for future genetic analysis. We are also planning to compare the predicted gene expression to measured gene expression to understand how the relationships between those two data groups might be utilized to better understand disease activity, therapeutics, and chronicity. And with that, I need to say thank you to all of the people who have been involved in this project, and especially to MPRINT. This project funding was essential for developing these JI EHR algorithms into something that becomes larger and that also integrates the genetic data that becomes very important for understanding relationships with therapeutics, which we are hoping to do in the future. With that, thank you very much.
Great, thank you, Dr. Patrick, for that presentation. I think we'll hold questions till the end so that we can hear talks from Dr. Antoon and Dr. Leech. So at this time, we will hear from Dr. Ashley Leech on maternal mortality and overdose in opioid-dependent individuals in pregnancy.
Right. All right, thank you. So hi, everyone. I am Ashley Leech. I am an assistant professor in the Department of Health Policy at Vanderbilt University School of Medicine. And thank you very much for the opportunity again to present an update on our work that really looks to examine maternal mortality and overdose in opioid vulnerable as well as opioid dependent pregnant individuals. Okay. So as a brief reminder of why we may care about this topic. So women in the US particularly obtain opioids at a much higher rate than men and also have double the growth rate in overdose deaths, an increase of around 600% in just 2 decades. Opioid overdoses in the past year, as of March 2022, have also contributed to over 100,000 deaths, and the highest being among those aged between 25 and 54, with an expected $2 billion in annual hospital costs related to overdose alone, not including healthcare costs for the disorder overall or for treatment. So we are undergoing 2 studies within this larger topic. The first study is a comparison of late maternal death and overdose among pregnant individuals with opioid use disorder compared to those without opioid use disorder, but possibly at risk or vulnerable for the disorder. So we will compare across 3 exposure groups. So the first exposure groups are those who filled at least 1 prescription for opioids, which is in the first box. Secondly, those who didn't fill any prescriptions for opioids. And then thirdly, those with either a diagnosis of opioid use disorder or have filled prescriptions for opioid use disorder within pregnancy. And it's important to note that these 3 groups are mutually exclusive groups. The second study that we'll be undergoing is really pulling from this 3rd group of pregnant individuals with opioid use disorder and examining the pharmacotherapy behavior among this group and the risk of maternal death and overdose. So for this presentation, I really plan on focusing on study 1, and then I will briefly describe study 2 when I review the next steps of the project. So the primary outcomes of the study include maternal death from any cause identified after delivery, mainly between 42 to 365 days within the postpartum period. Maternal death, so there has been a lot of work done on the proximal severe morbidity as well as mortality related to birth. And our study really complements, these other studies by assessing a period that excludes many of these deaths directly related to the pregnancy itself. Our other outcome is overdose diagnosis, both fatal and non-fatal. And then we will also be measuring really important covariates during pregnancy and at the beginning of the postpartum period, things like other mental health use or other mental health disorders, social vulnerability, things like that. The data source that we're using include Tennessee Medicaid data, which combines administrative claims data and enrollment data. And we supplemented this Medicaid data with the linkage to both birth as well as death certificate records for the state of Tennessee. Our study cohort comprises of individuals that have delivered an infant greater than or equal to 20 weeks between the periods of January 1st, 2007 to December 31st, 2019. And then we also narrowed our cohort to pregnant persons aged 15 to 44 years. So for our cohort breakdown, so we have a little over 1 million deliveries between 2007 and 2019. And then after, we got the deliveries between these periods of time, we then linked to the TennCare Medicaid recipients to their birth certificates, and then also linked the mom-infant dyads, which we have around 630,000 mother-infant dyads in our cohort. And then after, restricting on both age and gestation, we see that it really didn't significantly reduce our numbers in the cohort. And then for TennCare continuous enrollment requirements, so for our primary analyses, we are requiring that individuals are continuously enrolled for 90 days pre-delivery up to 41 days postpartum. And then for our sensitivity analysis, we're going to be requiring that individuals are continuously enrolled for 180 days pre-delivery up to 41 days postpartum. But for the purposes of our preliminary descriptive statistics, which I'll show you on the next slide, I will be focusing on the cohort highlighted in green, which are individuals continuously enrolled for 90 days pre-delivery and 41 days in the postpartum period. We are also excluding individuals with sickle cell disease because many of these patients will be on chronic opioids, but these individuals comprise, you can see, of just 0.1% of our entire sample. So we really didn't exclude very many people with sickle cell disease. All right, so moving on to our descriptive statistics over our 3 cohorts. So here you can see that the first cohort are those who filled at least 1 prescription opioid during pregnancy, which accounts for around 7% of our entire sample, or nearly 35,000 individuals. The second cohort, those without any opioid prescriptions during pregnancy, accounts for nearly 90% of our entire sample, or nearly 500,000 individuals. And then lastly, our third cohort, which is pregnant persons with opioid use disorder, account for 3% of our entire sample, or nearly 15,000 individuals. Going down, so the median maternal age at delivery is slightly higher among the opioid use disorder cohort. And then around 30% of Black individuals filled at least 1 prescription of opioids during pregnancy, compared to 65% of white individuals. However, we can see here, right, that most pregnant persons with opioid use disorder are white, which is something that we would expect. The number of prior deliveries are pretty evenly split across the groups, with the third group having a slightly higher lean towards prior deliveries. And then regarding route of delivery, we see slightly higher cesarean deliveries within the first cohort, which is something we would probably expect. And then among hospital births, the days of discharge may be slightly higher among pregnant persons with opioid use disorder. Pregnant individuals with opioid use disorder also have a slightly better social vulnerability score compared to the other two cohorts. So this scale of social vulnerability, 0 represents the lowest vulnerability and 1 represents the highest vulnerability based on their percentile position among all the census tracts in the US. And then further, for overdose within pregnancy, up to the 41 days postpartum period, we see very few overdoses. And then when we go to our outcomes, which is overdose within that follow-up period, the 42 to 360 day follow-up period, we can see that there are higher jumps across mainly the second as well as the third cohorts. And then when we look at maternal death from any cause, essentially we can see a higher proportion in the opioid use disorder group as well. And just to note, the less than 11 actually is just too small to disclose. So that's what we mean when we say less than 11. All right. So for our follow-up period, starting our tracking at day 42 post-delivery, we reported time to the loss of enrollment, time to death, which is not dependent on enrollment, and then 3 overdose, which is dependent on enrollment, but overdose death is not dependent on enrollment. So essentially these are independent events. And here we are left with individuals who remain in our cohort up to 365 days postpartum without having one of these other outcomes that we note. So you can see here that by the end of the period, you can see that around 111,568 individuals lost enrollment over the 365 days post-delivery, 383 individuals died, and 662 overdose from any substance. And we're actually in the process of separating between opioid use disorder, overdose versus other. And then 376,620 did not have any of these outcomes post-delivery. All right. And then in regards to our next step, so once we finalize our cohort for study 1, we plan to move on to the second study that will really assess outcomes within this third group only. So within this third group of pregnant individuals with opioid use disorder, either with a diagnosis or have failed a medication for the disorder during pregnancy. And so we will mainly be looking at, again, we'll be looking at maternal death and opioid-related overdose rates among pregnant individuals, both defined as persistently taking the medication during pregnancy compared to those who discontinued therapy prior to delivery. And then we'll also be looking at identifying risk factors for medication discontinuation, specifically things like medication type, medication switching, the role of dual substance use and mental health disorders, as well as things like social vulnerability. So that's the next step in our analysis. And this is a general timeline to complete both of our studies in terms of the analyses and the manuscript preparation. And that is all I have. Thank you.
Great. Thank you, Ashley. Very interesting study. Next, we will hear from Dr. James Antoon. James, are you able to share your slides?
I'm trying. All right.
Oh.
Can you guys see which?
Yep, that's the lecture view. Yep.
Like, this is the lecture view?
Yep.
Okay. Perfect. Thanks everybody for having me. I'm going to talk about my pilot project on Oseltamivir or Tamiflu use and a very specific drug-drug interaction. Don't have any disclosures. So we all know that influenza is a common and potentially serious illness. About 10% of children each year in the United States have medically attended influenza. There's only one FDA-approved treatment for children less than 5 now, and that's Oseltamivir, but it's by far and away the most common antiviral used. Over 99.9% of influenza antiviral use is Oseltamivir. And we know from lots of studies that Oseltamivir improves outcomes in children with influenza, but is underprescribed, especially in children who are at high risk for having influenza complications. And there's a number of reasons for that. And one of them is this black box warning about neuropsychiatric events in children, specifically suicidal ideation, psychosis, and seizures. But really this behavior change is the one that people most associate with these neuropsychiatric events. And this is challenging because influenza itself can cause neuropsychiatric events with influenza encephalopathy, and distinguishing between who's more likely to get Tamiflu-induced neuropsychiatric events versus influenza-induced neuropsychiatric events is very challenging. And so part of this project is to really try and identify who is at risk for these events and can we prescribe our antivirals accordingly. This is some preliminary work that we've done with TennCare to look at influenza-associated neuropsychiatric events. And it really, I think, gives evidence primed for precision medicine in that we can identify children who are certainly at much higher risk for these events with or without being exposed to oseltamivir. And those are children who are at high risk for influenza complications and those with psychiatric and neurologic comorbidities, which just so happens to be the group that's under prescribed Tamiflu. And we also know that we really don't prescribe as well as we could, and that these events occur at similar rates across those who receive oseltamivir and don't, where ideally you would see those who get oseltamivir have less events because you're prescribing it to children who are more likely to have influenza encephalopathy. And so the biological plausibility surrounding this is that oseltamivir passively diffuses into the brain and then gets rapidly exported out by P-glycoprotein. But in certain patients, the oseltamivir and its metabolites accumulate in the brain, bind to the NMDA receptor, which causes this cascade resulting in neuronal excitation, which is known to cause neuropsychiatric events in certain disease states, as well as adverse events in certain medications like ketamine. And so what we theorize is that potentially changes in P-glycoprotein might facilitate this increased oseltamivir in the brain in a specific pediatric population. And there's two ways this could be done. One is just genetic changes in P-glycoprotein, which we know can occur, but also certain drugs have drug-drug interactions involving P-glycoprotein. And that's where this project goes. And so the overarching theory is that kids who are on oseltamivir, if you get a PGP inhibitor, where you're not exporting oseltamivir out of the brain, you have higher levels, higher concentrations of oseltamivir, more of this NMDA GABA cascade, and more neuropsychiatric events. And so that's what we're exploring in this project, with the hopes of laying the groundwork of then moving down and looking at outcome events with these interactions. And these PGP modifiers are medications that are commonly used in kids. Things like dexamethasone and other steroids, PPIs, azithromycin, which is often misprescribed in influenza. And it's likely that children who are on oseltamivir, or at least some of them, are having these concurrent exposures, but we really don't know how often this occurs. And so the aim of this study was to determine, one, just what's the overall prevalence of oseltamivir exposure in children, and then two, how often are these drug-drug interactions or potential drug-drug interactions occurring? We're using the Tennessee Medicaid database as well. I won't go into why this is an ideal database for this, but I will say that Tamiflu in other national studies is highly prescribed in Tennessee. Tennessee is one of the highest prescribing states for oseltamivir overall in adults and children, and that's advantageous for this project because we're hopefully going to have a lot of kids who are getting exposed to Tamiflu. And just some preliminary results. We looked over 4 influenza seasons for children who had an ICD-10 diagnosis of influenza and found almost 200,000 kids had a oseltamivir prescription and an influenza diagnosis, and we had about 50,000 children who had oseltamivir dispensed, but no influenza, with durations that are suggestive of prophylactic use. So we can look at children who have treatment dosing and those who have prophylactic dosing. But doing this in a dichotomous fashion probably isn't the best way or most precise way to do it. And so when thinking about evaluating outcome events, we really want to get into exact measurements of exposure. And so for this project, we're in the process of assigning every day for each child gets a cohort assignment of being exposed to oseltamivir or not. And this is called assigning person time, which a lot of us don't do. So I'll talk a little bit about how this is done. So say we have 5 cohorts of exposure. One is not having an influenza infection and not having any medication use, so sort of the baseline nothing. Having influenza infection alone with no medication, having influenza and being treated with oseltamivir, which doesn't always start and finish on the same day. We think of oseltamivir as a 5-day acute treatment, but influenza being a 5 to 10-day illness. And so there's going to be a period after your influenza where you're still influenza positive, but you finish your treatment. And that period of time has a differential risk, or probably has a differential risk for influenza complications and oseltamivir complications compared to influenza alone or being actively on oseltamivir. And then you can get oseltamivir prophylactically with no influenza, which is a different dose and a different duration, and probably a different risk status. And so just walking through a couple example patients, you can have patient 1 who has influenza alone and no other exposure. So, for this 14-day period, they'll be assigned 4 exposure days to the baseline cohort and then 10 exposure days to influenza, knowing that 10 is about the maximum time that you think of influenza having its illness effects, at least in general, and we can vary this exposure time or definition as we want. Patient 2, on the other hand, might be assigned to a number of different exposure groups. That person might have 4 days of no exposure status, 1 day of influenza where they're diagnosed, and they pick up oseltamivir the next day, get their 5 days of influenza, and then have 4 days of this post-exposure period. So each person can contribute to multiple cohorts depending on what exposure they have that day, and you can assign a different covariate list for each person day. So this gets very, very precise in our assessment of both the exposure and the covariates. And then patient 3 might just have prophylactic oseltamivir, and have 10 days of exposure for that. So we can assign a number of different person days to each cohort for each potential encounter. And you can add differing layers of complexity to this, right? And so instead of just having these 5 cohorts, what if we assigned a dexamethasone and oseltamivir exposure group to try and assess one of these PGP modifiers. And these differing layers of complexity end up having multiple exposure cohorts. And this is just an example of 13 of them, but it gets even deeper than this when you start looking at comparator drugs and trying to look at drug-drug interactions for drugs that have similar indications but don't have PGP modifying effects. And so right now we're in the person day assignment phase of our project where we're going down each day for each person, assigning them a covariate list, assigning them an exposure day, and then we're going to add them all up and start doing some of our analysis. And so this is moving into a pretty exciting time where we're going to start generating quite a bit of data to assess these drug-drug interactions. And so that's all I have for today. Just thanking the group over here at Vanderbilt that's been guiding me through this. And that's it.
Thank you, Dr. Antoon. And with that, I will ask all of our speakers to join us. If there are any questions from the audience, please feel free to raise your hand and we can promote you to ask the question, or you are welcome to type it into the chat box. So I have a few questions I can start with while we're waiting. Dr. Patrick, so when you selected your number of ICD codes for the JIA to have, you selected 4 as the minimum. How did you arrive at that cutoff?
Yes, so in our cohort, when we looked at it, there was very small differences between 4 and 5 and some of these. And so one of the things we did is we looked at our cohort of 200 that we used to look at the PPVs, and then we also looked at the validation cohort. And between 4 and 5, the numbers really looked so similar, and we decided to go with 4 because at that point we had a PPV of 95, which we felt was like a really good PPV, and we also didn't want to exclude patients who we felt like the more numbers of codes we required, we were excluding patients. So it was more of seemed like the lowest possible number where we stopped gaining benefit from adding more.
Thanks. And then Dr. Leech, for you, and this is probably more of a clinical question for your Medicaid data. You said you were looking at patients who were enrolled for up to 41 days postpartum in the Tennessee Medicaid data set. How long does Medicaid typically last postpartum in Tennessee?
Yeah, that's a great question. So it changed in 2020, but it doesn't affect our cohort really because, well, I guess kind of like that's something that we are going to have to think about because we use data from 2007 up until 2019, and then we do have some follow-up right in 2020. But typically, I'm pretty sure it was around like 6 weeks. But now for 2020, it's been extended for up to a year, I believe. But that's a great question. It's definitely something that we're going to have to consider within those like 2020 follow-up individuals.
And then just for clarification, when the days of discharge you were talking about, was that for the mother or for the baby?
Mother. Yeah.
Thanks.
Mm-hmm.
Have you looked at baby data at all or plan to?
No, this is really focused on maternal outcomes, but I think in the future that could definitely be something that we look at. Yeah.
Great. Thank you. And then Dr. Antoon, on the Oseltamivir project, it looks like you're going to have a lot of different groups once you categorize your data. What's the follow-up statistics on that to see if there's differences between the groups? What's your control group look like, I guess?
Yeah, you use a modified Poisson regression in order to collapse all of your exposure days into your cohorts and then run your analysis. And so really the reference group will probably be untreated influenza, but it might depend on what the data looks like of what kind of makes the most sense with so many exposure groups. We'll probably collapse all of the inhibitors and inducers together, but depending if there's enough sample size, that may be too simplistic, and there may be some of these modifiers that are more powerful than others and might have a bigger risk. So depending on the numbers of these concurrent exposures, we might be able to isolate certain medications and look at their risk as well.
Great. I don't see any questions coming in from the audience yet. So with that, I will go ahead and close our session today. Thank you all for being willing to present your works in progress on these support pool fundings. And we will certainly look forward to seeing the final outcomes of the work. So thank you.